Okay, part two. The cells of the epidermis are produced by mitosis. If you've been in AP1 lab, you already know that mitosis involves making clones of cells. I'm just making more and more copies. So that mitosis is happening at the very bottom of your epidermis, the most deep layer that we have. And as new cells are formed, they push the older cells toward the surface where they slough off. Or if you want to be fancy, you can say disquamate. But pretty much as we divide the cells and they push upward, it's almost like a conveyor belt. Okay. Those cells that are getting further and further away from the base are basically getting to the surface where they can do the most protection. Keratinization. Again, if you look here, that's with a K and not with a C. Okay. So the cells will actually change their shape. See this delta here? Delta in my shorthand just means change. All right. So the cells change their shape. They change what's inside of them, their internal chemical composition, and they end up becoming filled with the keratin that I just talked about, being that kind of hard plasticky protein that we have. The cells eventually will die and produce the outer layer of dead hard cells that resist abrasion and form a permeability layer. The thing you have to remember about the way that your skin is, so here's that little scallopy edge. Our blood vessels are down here, right? We talked about the fact that the cells have to rely on diffusion to get things to and from those cells, yes? Well, as I'm growing and moving these cells further and further away from that base, as I get further and further away, I'm also getting further and further away from that blood supply. So, where I work in Alice, if you're from Alice, you'll know what I'm talking about, on campus. Right across from the parking lot is Burger King. So if I were to tell you, hey, go to Burger King and get me a burger, here's some money, you probably wouldn't put up a whole heck of a lot of stink about it, because I'm asking you to walk maybe the length of a football field to get me a hamburger. No, I'm not actually asking anybody to get me a hamburger, but you know what I mean. It's not that distant. Now, if on the other hand, I were to say, hey, I want you to go get me a burger, there's a Burger King in Dallas that I would really like to, but you can't take a car, you have to walk. I'm pretty sure you'd be like, no, right? The further that these cells get away from their blood supply there, the further away they're getting from their food. And as they start to move further and further up toward the surface of your skin, they kind of start to starve to death. So let's talk about the layers of the epidermis, okay? You have the stratum basale, also known as the stratum germinativum, according to your book, but I'll be honest, I've never heard it called that. I've only ever heard it called stratum basale. By the way, stratum is a very fancy way of saying layer, so it's the basale layer, okay? This is a stratum basale, and just like the name says, it is the most basal layer. It is going to be the one closest to the bottom. You then have the stratum spinosum. The stratum spinosum is the next layer up. After that, you've got the stratum granulosum. Stratum granulosum is the third layer from the bottom. Stratum lucidum. Stratum lucidum is only found in certain places on the human body, and we'll talk about that when we get into the details, but that would be the next layer up if it's present. And then the last layer, the one that we see, is the stratum corneum. So I want you to remember something, okay? Earlier, we talked about the fact that you've got these blood vessels down here. So these cells right next door are the ones that are going to be able to get rid of waste and to pick up nutrients. The further I go up this direction, the further I get away from this blood supply. Okay? Just keep that in mind. So let's start with talking about this stratum basale. Okay? It is structurally the deepest layer of the epidermis. It's a single layer. You can see the color difference between this basale and the spinosum. It is a single layer of cube to columnar shaped cells. And this is what attaches to this basement membrane. Again, you can see the basement membrane in red right here. Okay, so that's that double-sided sticky tape. I want you to notice something else. See these things? Remember we talked about those special cell-to-cell connections in chapter 4? These are the hemidesmosomes holding my stratum basale cells to that basement membrane. Okay? So, functionally, what is happening with this layer? This is the cell layer that actually does the mitosis that creates all of these cells that are coming up toward the surface of our skin. Okay? So literally, when we talk about what's the origin of the skin, this is the origin of the skin that you see. All of the cells that are up here started down here. Okay, and like I said, it's kind of like a conveyor belt. I divide my cell in two, one goes up, the other stays at the base. Now, I'm going to go back just for a second. See this cell right here? That one's in metaphase. When we talk about the division of the cells, one of the cells is going to stay there, one of them is going to stay here in the base, and then the other one is going to get pushed toward the surface. So, one of the functions of the stratum basale is I produce the most superficial layers of my skin. The cells come from here. The other thing that you find in the stratum basale are the melanocytes. Now, we talked about the fact that the melanocytes contribute to your skin color. So the melanocytes that are in this layer are actually contributing melanin into cells. Melanin protects from UV light. And again, we're going to talk about that in a bit of detail in a little bit. So I mentioned the hemidesmosomes being present. The desmosomes are also present. If you look, see these little green guys right here? These are going to be the desmosomes holding the cells to their neighbors. And you can actually see them up here holding the cells to their neighbors. Okay? Keratinocytes, those specific cells, undergo division every 19 days. One daughter cell, like I said, gets pushed to the surface, while the other stays in the stratum basale down here. It takes about 40 to 56 days from start at the stratum basale to sloughing off. So every 40 to 56 days, every about 2 months, you have kind of completely new skin. The turnover to get all of those cells sloughed off and new cells put in place has happened. Now, that doesn't mean every 60 days you go to sleep and your skin falls off and then you wake up with new epidermis. No, it's just the turnover rate for start to finish. Okay? So, let's talk stratum spinosum. That is going to be this layer right here that I don't know why that wasn't writing. That layer right there. So this kind of orangey looking layer. It's about 8 to 10 layers of many-sided cells, and they flatten as they get further and further toward the surface. Because by the time that we get up here, they're pretty pancaked up, right? So as we push further this direction, these cells kind of go from being cuboidal or columnar to getting more squished, more compressed, okay? Now, there's kind of a cost to that. Remember those desmosomes that I was talking about that are holding the cells to their neighbors? Yeah, I actually have to break those down when I change the shape of the cell and then reform them. So, the best way that I can think to explain this, imagine I have two students in front of me standing side by side, and I tell them to raise their arms and like put their hands together. So, literally from foot all the way to their hand, they're touching each other. Just go with me on it. Okay? Now, imagine I super glue those two students together. So from hand all the way to foot, their sides are super glued together. Now, let's say that I have one student and I will not let that student squat down, but I start pushing the second student down, even though they're glued together. What's going to happen? Eventually, the skin would tear, right? So I can't do that. If I'm going to change how they're kind of standing next to each other, I either have to let them both go the exact same way, or I kind of have to dissolve the glue, kind of reposition them, and then glue them back together again. So these desmosomes that you can see here holding these cells together, as the cells start to get squishier and flatter coming up this direction, I kind of have to break them down. And then once they're in a better shape, or in a flatter shape, I shouldn't say better, they reform those desmosomes. So why do we call it the spinosum? Well, when you have cells that are being held together by desmosomes, remember that you've got those two discs and those intermediate fibers that are kind of anchoring through both of them? When scientists decided that they wanted to try and split these cells apart microscopically, and they started to pull them apart like you're trying to, pull apart, I don't know, a ribbon, or you're trying to pull apart thread, what they ended up with were cells that kind of looked like this. They look spiny. And one of the reasons was because, remember those desmosomes? Well, they're still trying to connect to their neighbor. So when you pull this apart, you end up with cells that look spiny. So they called it the stratum spinosum specifically because of that. Okay. Something else that's happening here, we have additional keratin fibers being added, and we have these lipid-filled organelles called lamellar granules. So if you look here, see the fibers, these long pieces? Those are going to be additional keratin fibers. And then see these kind of red bubbles here? These red bubbles are lipid-filled lamellar bodies, okay? And I know you're probably thinking, okay, well, that's not too special. It'll become special in a minute, okay? So what's the function of this layer? What's the function of the spinosum? One is to produce those extra keratin fibers. Why? Because I need the keratin to protect my outer body. So making extra in this layer as it gets further toward the top of my epidermis makes sense. Now, the other thing is the formation of these lamellar bodies. We do actually need these lamellar bodies. Okay, so again, I'll explain why in just a second, but it is something that is required. It isn't optional. I'll put it that way. So the next layer up is this one, the stratum granulosum. Now, the stratum granulosum is called the stratum granulosum because when you look at it under a microscope, it looks like you've got grains inside the cell. See the little purple spots here? Even when you look at an actual skin cell slide at the granulosum, you will see that kind of same, it almost looks like somebody put purple sugar there, okay? So you've got 2 to 5 layers of these flattened diamond-shaped cells. That's the structure, okay? They produce keratohylan granules. Keratohylan is the thing that stains that makes it look like it's grainy. Keratohylan is again one of those, it's going to make the cell hard and able to resist abrasion proteins. Remember those lamellar bodies from earlier? They actually get released from the cells. See these little yellow globs here? Yes? Those are the lamellar bodies. So when you talk about having dry skin and you put lotion on your skin, you're basically putting oil on your skin. When you talk about having dry hair and you put conditioner in your hair, you're putting oil in your hair. So when we talk about these lamellar bodies having lipids, it's oil. One of the other events that happens between the stratum spinosum and the stratum granulosum, this is our transitional layer. At this layer of our epidermis, we have gotten so far away from this blood supply that we start to die. These cells start to starve to death. So if I've got dead cells and I don't want them just to kind of slough off, I need to moisturize them. So the lamellar bodies getting kind of released at this layer helps to kind of moisturize the cells that are starting to die. So the cells start to die, like I said, in this layer. The nucleus and the other organelles start to break down. They start to degenerate. But those keratin fibers and the keratohylan granules stay intact. This is what gives us that hard layer of cells. Okay? Now, let's talk about the next layer up, the stratum lucidum, which you can see right here in this green color, although why it's green, I don't know. Let me explain why I say that. Okay, lucid. The word lucid, if you have a lucid patient, it means that they have a clear head. They know what's going on, they know who you are, they know who they are. Lucid means that it's clear. The stratum lucidum is a clear layer. That's why they call it the stratum lucidum, because even when you're making slides, a lot of times this will not stain, okay? It's a 3 to 5 layer, 3 to 5 layer stratum in your skin with no distinct boundaries. The cells are so tightly compacted together that you can't even really see what a cell is. You just know it exists there. That layer exists there. And the thing is, it's so tightly packed with the keratin and the keratohylan that the dyes that we use, the stains that we use to stain slides, won't penetrate it. So when you're looking at a slide of skin that has the stratum lucidum, you don't see anything there. It just looks like a clear line, like there's nothing there. So that's one of the reasons why we call it the stratum lucidum. It appears transparent, and normally this is only found in thick skin. So let's talk thick versus thin real quick and real surface so that we can move on, but then we'll get into it in, I think it's another couple slides. Thick skin is what you have on the sole of your feet. It's what you have on the palms of your hand. This type of skin is a lot thicker than the rest of the skin on your body because we walk on our feet and we use our hands to grab things. We need them to be even more protective than the rest of our skin. Everybody's done that thing where you take a needle and put it through, your finger and it's just in the surface layers. So, but you go, right? Okay, everybody does this. Do it on the back of your hand. Yeah, you're not going to, because that would be painful, right? So on the back of your hand, you lack that stratum lucidum. It is not thick skin. So why do we have it? Well, it actually is used to disperse and basically compact the keratin and the keratohylan fibers that we have. When you think about the bottom of your feet take a lot of abuse. We walk on them, every day. So, with that in mind, we need to have kind of that extra layer of protection where we have the extra thickness and compacted cells that are holding the keratohylan and the keratin fibers so that we don't damage them. Okay. Yes, we can go and we can get a pedicure and they can do the whole shavy foot thing that they do, but ultimately they're still not taking everything. Okay. So the last layer, the stratum corneum, which is up here. Okay.